Okay, we'll have a quick little video here about cell theory. So we're going to see that when we're talking about cell theory, we're talking about very small things. Cells are very small, micrometers. Before cell theory, before knowing about the very, very small things in this world, people had superstitious ideas about what caused disease, because they didn't know about the microscopic world, so they had all kinds of strange beliefs. So you're talking about something so small, we're going to need the help of an instrument like this. And this is the light microscope. This one looks pretty impressive and pretty modern. However, the one I'm going to show you a picture of the one that was used by a man called Robert Hooke, who was the first one. A guy who first observed the cell, and he is the origin of the word cell. He came up with the word cell. And the reason why he came up with the word, you could see these boxes, alright? You could see boxes, or you might think of cells. When I think of the word cell, yeah, I think of cells like living cells, but I also think of a prisoner's cell. And for Robert Hooke, he looked at these boxes and he thought they looked like the cells that monks lived in. So monks at the time must have had their own cell or their own small room. So he saw these, and he saw these cells on cork. Cork, which is the outer parts of a tree, I believe. Types of microscopes. So yeah, the one that he would have been using. There's a light microscope, and it was invented in the 17th century. So Robert Hooke and von Leeuwenhoek would have been, or whatever his name was, would have been using these kind of these would be the microscopes of the time. It'd be a single lens. Light microscopes, I guess this must be referring to today, but they can get 1,500 magnification. But I guess that's today's microscopes, not the microscopes of the 17th century. And now we've got the problem with light microscopes is you cannot get really, really good magnification. With electron microscopes, and here we can see a transmission electron microscope, you can get 500,000 times magnification, which is incredible. And you can see that looks like a sophisticated piece of kit there. And that is the transmission electron microscope. Good for studying internal structures of cells. Alright, so maybe it looks like it takes sort of cross-sectional views of the cell. Now I have to say that instead of using light, the electron microscope uses electrons. And I think they also use a probe. Light has a wavelength of 380 to 750 nanometers, so you're never going to get good detail, the kind of detail you would want with light microscope. But there is this second transmission electron microscope. Another kind of electron microscope is a scanning electron microscope, also invented at the same time as the transmission electron microscope. And the scanning electron microscope is good for looking at the surface of bacteria, viruses, and tissues. And here we have a picture of a very small worm, I think, with the scanning electron microscope. And here on the left we can see photos taken by scanning electron microscope, and you can see the 3D structure, the surface of what looks like bacterial cells, and oh no, actually I think they're pollen grains. Oh yeah, it says they are pollen grains. There are pollen grains. I totally said the wrong thing. Pollen grains on the left, and the same pollen grain, I suppose, is on the right. And what is this? A cell? Is a pollen grain a single cell? Good question. I should have found the answer to that before I made this video. Alright, but yeah, you can see. I guess the only thing is to see the beautiful detail here and how the usage. Scanning tunneling microscope. Now, can this be any more impressive than, because it can get right down to the level of atoms, and we can see the atoms on the surface of, I don't know what this is the surface of, but it's the surface of something, and you can see the individual atoms. So, cell theory. These are the tenets of cell theory. First one being all organisms are made of one or more cells. Alright, yeah, so maybe I should say Von Hooke saw cells in cork. But then you have the other guys, like Schwann, picture there, and Schleiden, who they were looking at plant cells and animal cells, and they saw the same thing. These little boxes, these cells, these little cells that the living things, plants and animals, seem to be made of. So I guess it got them thinking, hey, I guess this could be a feature of all living things, that they all have a cell. And so number one is, yeah, that is it. All organisms are made up of one or more cells. Yeah, it could be one cell. For example, there are eukaryotic cells that are only one cell, and of course, prokaryotic cells, which are only one cell, and I'll mention the difference between the two soon. Two cells are the basic building blocks of life. Yes. Okay, so number one and number two fairly, fairly similar, right? Fairly similar ideas. And number three, all cells come from existing cells, right? So they don't just appear out of nowhere, they come from another cell. So I guess number three is true for every cell that exists today, but perhaps at one time, back billions of years ago, it's not true for the very first cell. Okay, number three. Oh yeah, and so just to finish, I just want to say that to mention the difference between a eukaryotic cell and a prokaryotic cell. A eukaryotic cell has internal membranes, so as in some organelles, which are themselves got membranes around them, whereas a prokaryotic, and I think the nucleus is quite an important organelle in the eukaryotic cell, whereas the prokaryotic cell does not have internal membranes, it does not have distinctive organelles. DNA is sort of floating around. I don't know if it's floating around, but it's circular. It's circular. It's not organized to the tight nucleolus like you got in the eukaryotic cell. And we can see here this prokaryotic cell has some cilia that help it probably propel through the medium, the water that it's in. See you later.